Hello friends, so in this video I'm going to talk about the 20 common amino acids of protein. The structure formulas show the state of ionization that would predominate at pH 7.0. The unshaded portions are those that are common to all the amino acids, just like that. These portions are common to all the amino acids, and these are the R portion, and the shaded portions are the R groups. Although the R groups of histidine is shown R charge, its pKa. Where is the histidine? Here is the histidine. Histidine is the positively charged R group, and group of histidine is an uncharged in pKa, which we will discuss later. In such that a small but significant fraction of these groups are positively charged at pH of 7.0, and the protonated form of histidine is shown above in the graph, which we will discuss later. The first is non-polar aliphatic R groups. The R groups in this class of amino acids are non-polar, hydrophobic. Non-polar and hydrophobic, the side chains of alanine, valine, leucine and isoleucine tend to cluster together within the proteins, stabilizing protein structure. Their function is to stabilize protein structure through the hydrophobic effect, and lysine has the simplest structure. Lysine is the simplest one amino acid, and although it is most easily grouped with the non-polar amino acid. Methionine, one of the two sulfur-containing amino acid. Methionine is a sulfur-containing amino acid and has a slightly non-polar thioether group. Methionine has a non-polar thioether group in its side chain or R chain. Next is proline. Proline has an aliphatic side chain with a distinctive cyclic structure. The secondary amino acid, that is amino group of proline residues, is held in a rigid conformation that reduces the structural flexibility of polypeptide regions containing proline. Next classification is aromatic R groups. Phenylalanine, it includes phenylalanine, tyrosine and tryptophan, with their aromatic side chains are relatively non-polar and hydrophobic. All can contribute to the hydrophobic effect. The hydroxyl group of tyrosine can form hydrogen bonds. This is a very important point, that the hydroxyl group of tyrosine can form hydrogen bond, and it is an important functional group in some enzymes. Tyrosine and tryptophan are significantly more polar than phenylalanine, because the tyrosine hydroxyl group and the nitrogen of the tryptophan endorse ring. Another most important point is that the tryptophan and tyrosine, and to much lesser extent phenylalanine, absorb UV light, that is ultraviolet light. This accounts for the characteristic strong absorbance of light by most proteins at a wavelength of 280 nanometer. A property exploited by the researchers in this characterization of protein. Now, this is the graph that shows the absorption of ultraviolet light by aromatic amino acids. You can see here the comparison of light absorption spectra of the aromatic amino acids: tryptophan, trypsin and phenylalanine at pH 6. Here. The amino acids are present in equimolar amounts under identical conditions. The measured absorbance of tryptophan, this is the tryptophan in black color. The measured absorbance of tryptophan is more than 4 times that of tyrosine. Yes, this is the tryptophan and this is the tyrosine in red and phenylalanine in blue color. Note that maximum light absorptions for both tryptophan and tyrosine occurs near 280 nanometer. This is the peak where absorbance of these amino acids at 280 nanometer takes place. Light absorption by phenylalanine generally contributes little to the spectroscopic properties of proteins. Next class is polar uncharged R groups. The R groups of these amino acids are more soluble in water, hydrophilic or more hydrophilic than those of the non-polar amino acids because they contain functional groups that form hydrogen bonds with water. This class of amino acids includes serine, threonine, cysteine, asparagine and glutamine. The polarity of serine and threonine is contributed by their hydroxyl groups and the polarity of asparagine and glutamine is contributed by their amide group. Cysteine is an outlier here because its polarity contributed by its sulfhydryl group and it is quite modest. Cysteine is a weak acid and can make weak hydrogen bonds with oxygen and nitrogen. Whereas asparagine and glutamine are the amides of two other amino acids also found in proteins, aspartate and glutamate, respectively, to which asparagine and glutamine are easily hydrolyzed by acid or base. Cysteine is readily oxidized to form covalently linked dimeric amino acids called cysteine, in which two cysteine molecules or residues are joined by a disulfide bond. This is the important property. The disulfide linked residues are strongly hydrophobic, non-polar. Disulfide bonds plays a special role in the structures of many proteins by forming covalent links between parts of a polypeptide molecules or between two different polypeptide chains. Positively charged basic R group. The most hydrophilic R groups are those that are either positively charged or negatively charged. The amino acids in such R group have significant positively charged at 7.0 pH. Are lysine, which has a secondary primary amino group at the epsilon position on its aliphatic chain, and arginine, which has a positively charged guanidinium group. You have to remember that group, that is guanidinium group, and histidine, which has an aromatic imidazole group. You have to remember the groups they contain as the only. The next is negatively charged acidic R groups. The two amino acids having R groups with a net negative charge at pH 7 are aspartate and glutamate, each of which has a second carboxyl group. There are also uncommon amino acids which have important functions. In addition to the 20 common amino acids, protein may have contained residues created by modification of common residues already, in which number one is selenocysteine and other is pyrolysin, are special cases. These are rare amino acid residues and are not created through a post-synthetic modification. Instead, they are introduced during protein synthesis through an unusual adaptation of the genetic code, that is UGA, which we described early in the video. And then selenocysteine contains selenium rather than sulfur to cysteine. They are not sulfur containing amino acid, but they contain selenium rather than sulfur. Actually derived from serine, selenocysteine is a constituent of just a few known proteins, and pyrolysin is found in few proteins in several methanogenic means methane producing bacteria and archaea. It plays a role in methane biosynthesis, and some amino acid residues in a protein may be modified transiently, and they alter the protein's function. In addition of phosphoryl, methyl and acetyl, adenylyl and ADP ribosyl, other groups of particular amino acid residues and can increase or decrease protein activity. Phosphorylation and it's a particularly common regulatory modification. Other is ornithine and citrulline. Deserve a special note because they are key intermediates. Ornithine and citrulline are key intermediate metabolites in the biosynthesis of arginine and in the urea cycle. So these all are the rare amino acids that are cysteine, pyrolysin and ornithine and citrulline, which also have a spatial function, that is ornithine and citrulline included in the they are the key intermediate and metabolites in the biosynthesis of arginine and in the urea cycle, while selenocysteine and pyrolysin are special case that they are adapt from genetic code, usually A, and selenocysteine contains selenium rather than sulfur or cysteine, and pyrolysin is found in few proteins in several methanogenic producing archaea and in one growing bacterium, and it plays role in methane biosynthesis.